So for everyone who's joining, we're just going to give it an extra 5 minutes, just to allow for some people to join. We should start at 5 past.
Dylan, if we've lost 5 minutes, I can speak a bit more quickly. Don't worry. That's
Okay.
fine. No problem.
Okay, I'll start with giving the introduction, and I'm sure more people will join as the seminar gets underway. So firstly, thank you everyone for joining. We have a very interesting seminar today by Professor Stephen Allen on the role of gut health in childhood malnutrition and development. So Professor Allen is currently a professor at the Liverpool School of Tropical Medicine, and he'll be leading the seminar for around the next 40-ish minutes or so. He is currently the Secretary of the Commonwealth Association of the Pediatric Gastroenterology and Nutrition, otherwise known as CAPGAN. And a lot of his research has focused on pediatric gastroenterology with a focus on interventions to improve gut health across a broad range of conditions, including malnutrition and inflammatory bowel disease. He also holds the position as one of the gut health workstream leads for the Action Against Stunting Hub, and here he is currently evaluating the interrelationships between gut health and gut microbiota, epigenetics, and also evaluating an early life synbiotic study in Senegal. And he's also currently working on a similar study of early life supplementation in Western Kenya. So without further ado, I will hand over to Professor Allen.
Thank you very much, Dylan, for that very kind invitation. I'll just share my slides if I can. Okay. Does that work okay? Can you see my slide?
Yeah, we can see them.
Okay, so thank you very much for the invitation to talk on this topic. I'll be talking about focusing on gut health and trying to explain what that might mean in early life and how that contributes to childhood malnutrition and particularly poor child development, and how we might harness gut power to improve growth and developmental outcomes and possibly even survival for children in poorer countries. As you said, I'm based at the Liverpool School of Tropical Medicine, but a lot of my work recently has been in Western Kenya, funded through the Children's Investment Fund Foundation and working with KEMRI. I know that Iwareta Titi is on the line from Kenya and who delivered this study, the PROSYNK study, which I'll be talking quite a bit about. And as you said, also involved in the Action Against Stunting Hub, looking at very similar issues across Senegal, India, and Indonesia. So very briefly, I'm going to try and start with a general sort of good early start in life and the importance of that. And then, as everybody on this call, I'm sure, is familiar, the persistence of poor growth and development in early life in poorer countries. And then how poor gut health really captured as this term environmental enteric dysfunction, contributes to that and how it might provide us with a new opportunity. And then much of my talk is going to focus on what we've found in an intervention study with probiotics, synbiotics in Kenya. That's the PROSYNK study. I'm going to talk a little bit about what we hope the Stunting Hub will contribute to this area. And then a couple of slides just on how we might thinking about developing this gut power concept going forward. Our results from Kenya are still fairly new, and we're still thinking about what they mean. So I hope we can have an open discussion, and people can contribute their thoughts and what they think, how we might take things forward. So this business of a good early start, this is a position paper from the Academy of Medical Sciences in the UK, trying to advocate to the government in the UK to prioritize early childhood. So this is clearly an international endeavor across many countries that we should optimize the early growth and development of children. This aligns with the developmental origins of health and disease, where we know that poor growth, adverse events in early life have long-term impact on the life course. So the impact of various factors during early years is profound and long-lasting, since this is a major period of brain and organ development, and interventions in childhood are likely to be much more effective about preventing chronic diseases such as cardiovascular disease, stroke, hypertension, type 2 diabetes. They're much more likely to be effective early in life than later. And this is a UK perspective. Of course, we translate that into the first 1,000 days in poorer countries, which I'm sure everybody is familiar with. What I want to do as well is focus on chronic systemic inflammation in the blood, as a critical disease mechanism that seems to be the issue that mediates this poor early start with poor outcomes. So just picking off three major topics, we know that almost half of children in poorer countries fail to reach their developmental potential, and we know that's very much associated with systemic inflammation, that is associated with brain connectivity and future cognitive outcomes. And really maximizing children's developmental potential in terms of brain development is probably the key issue that we should all be focusing on and thinking about. More important than a few extra centimeters in height, obviously it's that whole development, including the brain, that's important. But of course, inflammation is a major pathway to stunting. We know almost 1 in 5 of the world's children remain stunted. And stunting is characterized by chronic inflammation in many different studies, and indeed is thought to mediate some of the poor growth, and we'll talk about that a bit later. And then perhaps equally importantly, we know that the main killers are non-communicable diseases in adults. Most of those deaths occur in poorer countries, and as we've said already, it's chronic inflammation throughout the lifespan that underlies the development of chronic diseases. So if we can target chronic systemic inflammation, whether or not we can do anything about growth and stunting, we would probably ameliorate much of the adverse outcomes associated with poor growth in early childhood. So now I want to focus on the gut, because I'll put it to you that the gut is the main organ that is responsible for this chronic inflammation in children in poorer countries. So this is a healthy gut mucosa, at least from a Western perspective. This is what we expect to find in healthy children. I'll just focus on this sort of picture at the bottom right. Clearly, if you take a biopsy from the distal duodenum, the beginning of the jejunum, where most food digestion and absorption occurs, we expect to see very long finger-like villi, and together with microvilli, which you can see in this electron microscope, on the surface of these villi, these really magnify the internal surface area of the small intestine. So what on the outside is a sort of short hose pipe, if you unravel it and spread out the surface area on the inside, it's equivalent to a double tennis court. So that huge surface area is needed to digest and absorb food, nutrients, but of course has to be protected. And what you can also see from this histology section is within the lamina propria here, the tissues of these villi and underlying the villi, all these black dots are immune cells, lymphocytes, neutrophils, macrophages, a whole collection of different immune cells. And in fact, most of our immune system is in the gut. 60% of our immune cells are in the gut, and they are separated from all the bacteria, other organisms, food antigens, etc., that are in the gut lumen that we take in by mouth every day, only by the mucosa. But these mucosal cells are healthy. They're standing shoulder to shoulder, tightly linking arms, and it's a highly regulated communication between the gut lumen and the immune cells in the tissues. I'll come on to that in a second. What you can also see in this picture is down here are the crypts, and this is where new intestinal cells are made, the mucosal cells. So these crypts are gently working away, producing new mucosal cells that migrate up the villi and then fall off at the end, probably every 24, 48 hours or so. So that is what we think a healthy gut should look like, which is on the left here. The picture on the right here is the gut biopsy from a child in Zimbabwe from Paul Kelly and Beatrice Amadi's group with stunting that did not respond to a supplement. And you can see immediately, this is not as healthy as the one on the left. So there's subtotal villus atrophy, the villi are blunted and broadened. So that surface area has radically reduced. So it's not surprising that if all you do is feed these children, they're not necessarily going to respond because they just don't have the gut surface area and the healthy intestinal cells to benefit from being given nutrients. Also, there are breaks in the surface, and that's not only where the mucosa has been completely eroded, but also leakiness in the actual mucosal cells. They're not standing shoulder to shoulder, tightly linking arms. There are gaps between them through the so-called tight junctions. So that means all the microbes in the gut and the food antigens have much easier access to the child's immune system, so are constantly causing inflammation. And you can see much greater inflammatory infiltrate in this mucosa, and indeed that translates out into the blood. You can also see that the crypts here are much longer and are probably working harder. They're trying to make new mucosal cells to repair these villi, but because of the ongoing inflammation, they're losing the battle. But they're consuming a lot of energy trying to repair the villi, and that energy should, of course, go to growth. That's a moderate lesion. This is a severe lesion. So this is a child, this is just the severe end of the spectrum, would be very common in children with severe malnutrition. And you can see here, nearly all of the mucosa is crypts, trying to repair the mucosa. The villi have almost completely lost, and there's large areas where the barrier has completely broken down. This sort of insight into what's going on in the gut requires a biopsy, which usually requires endoscopy and general anesthetic, which is clearly not appropriate for field studies. So what we do is use biomarkers, and the three biomarkers I'll talk about are probably the ones that have been used most commonly in malnutrition studies. Myeloperoxidase in the stool. Myeloperoxidase is in immune cells, the cytoplasm of immune cells. If they're in greater number and they're leaking out into the stool, myeloperoxidase goes up. So it's a marker of intestinal inflammation. Alpha-1 antitrypsin is a protein made by the liver, a kind of anti-protease protein. So it has a defense against infections and oxidant stress. It's made in the liver, but it's supposed to stay in the blood. It is not supposed to leak out into the gut lumen and into the stools. So if alpha-1 antitrypsin goes up, that's a marker of increased gut permeability. And then the third marker that we've used is intestinal fatty acid protein. That's in plasma, not in stool. And that protein is pretty much restricted to the mucosal cells of the proximal small intestine. So if they are stressed or damaged, then that leaks through into the bloodstream and is a blood marker of mucosal damage. So those are the three biomarkers I'll be talking about. But it's this is just sort of the structure and function of the bowel. So that's one thing, but clearly the situation is much more complicated. So here's a fairly old picture showing the very active immune interaction between microbes in the gut of all different shapes and sizes, and then all these immune cells that are sitting in the lamina propria. So sitting over Peyer's patches, which are collections of immune cells, we have specialized mucosal cells, M cells, and they're constantly sampling the gut contents. And with the help of dendritic cells, here's a dendritic cell that's reaching out and tasting a bacteria. Here's one that's ingested a bacteria and is bringing it into the gut to present to the immune system. And here are the CD4 cells that then drive B lymphocytes and T helper cells and the likes. So both cellular and humoral immunity. So this close interaction is going on in all of us all of the time, but in general, our immune system is recognizing bacteria in the gut, but it's tolerant. It's calming down and preventing an inflammatory response like you've seen in those slides of environmental enteric dysfunction. So there's a very complex interplay on the immune system side, as well as just structure and function that we measure with the biomarkers. And then to take that even further, and thanks to UKRI for finding this very good picture, this is a proposed model of how the gut enteric infection, malnutrition, causes systemic inflammation and therefore affects the brain through the gut-brain axis. So this is a hypothesized mechanism. I don't think anybody knows how systemic inflammation leads to these long-term adverse outcomes, but it's probably along these lines in here, particularly for the brain. So it's about dysbiosis, gut bacteria leaking through the tight junctions, stimulating inflammatory and immune responses. So you can see here in pro-inflammatory cytokines. These then impact on the brain microglia, which is crossing the blood-brain barrier, and impair brain development. And also here's a malabsorption component to that. So in fact, although we measure biomarkers in the gut, we should perhaps be measuring a whole range of different biomarkers related to poor gut health. For example, biomarkers of microglia in the brain. So biomarkers are organs quite distant from the gut, because that is how we think these impaired developments and chronic diseases develop. Okay, so what we've been testing in Senegal, in the Stunting Hub, and in Kenya in the PROSYNK study is this, I think, very well accepted and very well documented pathway. We know that adverse environmental factors, particularly poor sanitation and hygiene, but also exposure to antibiotics, cesarean section delivery, poor feeding practice, this perturbs the gut microbiota, even in very young babies who are exclusively breastfed. And this results in enteropathogen colonization. We know from the malady studies and other studies that we get pathogens entering into the gut, even in very young breastfed babies. They don't necessarily cause diarrhea, but they adhere to the gut mucosa and cause the lesion that we've been talking about. And the elements of this are atrophy, so a loss of surface area, inflammation in the gut, and then increased permeability. This has two major outcomes. One is systemic inflammation in the blood because of the leakiness in the gut. And we know, as we've been discussing now, we know that that's a mediator of poor outcomes and also growth hormone resistance, so directly impairing the action of insulin-like growth factor 1 at the growth plates. And then there's also a malabsorption element. So both of these factors contributing to poor growth, but also poor development. We hypothesize that if we gave healthy bacteria, so-called, so probiotics and symbiotics, if we boosted healthy bacteria in the gut, and the types of organisms that we know dominate in the healthy breastfed infant, so bifidobacteria and lactobacilli, if we boosted those, could we strengthen the gut's resistance against colonization to enteropathogens? Clearly, if there's a very healthy gut flora, it's more difficult for pathogens to colonize. And also, they will also do direct things in the gut. They will directly improve the gut mucosa integrity. They will probably stimulate immune responses towards tolerogenic responses. They will directly reduce inflammation. So there are numerous mechanisms by which they may be beneficial. Will this lead to a healthy gut? We wouldn't have these pathways and we'd have a healthy child. So that's the hypothesis we've been testing. So I want to now switch to focus on the PROSYNK study. I should pause at some point. If I can't see anybody on this, so if anybody's got any questions, please just shout, and I'm very happy to stop at any point. So this is the study that Iwaret and her team have been running in Western Kenya, in Homa Bay County, in a rural population, mainly rural population, clearly quite poor with a lot of stunting and malnutrition. And what we did briefly was a randomized 4-arm open study. So we took newborns, 600, and randomized them to receive one of 3 different pro-symbiotics, because we didn't want to put all our eggs in one basket with just one product because of the issues of strain specificity. So we thought, let's just try 3 different products, and then controls who did not receive any intervention. And the children were dosed with these probiotics or symbiotics daily for the first 10 days of life, and then only weekly up to 6 months of age. And then intervention stopped, and we followed them up at 1 year, and we've also followed them at 2 years. Although the controls didn't get any symbiotics or probiotics, they still got the visits by the research staff. So they were visited daily for 10 days and then weekly to 6 months. And this design, I think, is very critical to understanding what we found in this study. Here is a lady who's a peer mother. She's a lady in the community who's had a low birth weight infant herself. Sorry, my pointer's disappeared. Here she is. She's had a low birth weight infant herself in this community. She lives in the community, and she knows about caring for small, vulnerable babies. This group of peer mothers was set up by Helen Nabwera, who was in Liverpool and is now moved to Kenya. So we utilized this group, and they were the ones that supported really their neighbors in administering the interventions, but also they gave advice about hygiene, healthy infant feeding, breastfeeding, and also if the child was sick, then they helped the mother to facilitate a referral. And indeed, we had doctors employed in clinic who would look at these children from our study, even during times of strike, and they had their own supply of medicines and what have you. So that's how we run the study. Although this was an open study, all the laboratory biomarkers were done blind. So the laboratory staff had no idea which group these samples were from. So we're very happy that the results are not biased by being from an open study. Our primary outcome was chronic systemic inflammation. That was assessed by plasma AGP, a marker of chronic inflammation in the blood, alpha-1-acylglycoprotein. But then we also measured acute inflammation in the blood, CRP, and then the biomarkers of intestinal health that we've talked about, growth hormones, and we looked at morbidity and mortality. These were the interventions. They're off-the-shelf interventions. The Labinic is used a lot for preterm babies throughout Europe, certainly in neonatal units to prevent necrotizing enterocolitis. But we had that formulated for our study as a symbiotic with the addition of fructo oligosaccharides. And then the proven 4B, the Lab 4B probiotics, we use that as just the probiotic organisms, and then also formulated as a symbiotic with fructooligosaccharides. These come as powder in capsules, and you can see they're slightly different colors in this open study. And the dose was 1 capsule a day, give the powder to the baby. That's daily for 10 days and then weekly to 6 months. So the study went extremely well. We enrolled fairly quickly. This is a population with a high level of poverty, 64% rural, 18% of the mothers were HIV positive, but as far as we know, none of the babies seroconverted to HIV. And we did baseline demographic clinical wash variables, and they were all very similar. So we feel that the only differences between the study arms is whether with the interventions. They were very similar for all the other variables. So the randomization appears to have worked. So what did we find? This is our primary outcome measure, and this is our kind of headline result. So this is blood plasma alpha-1 glycoprotein. So this is a marker of chronic inflammation. And this level 1 here is the upper limit of normal. So if you're above this line, you have chronic inflammation in the blood. And you can see this at these levels at 6 weeks, 3 months, 6 months, at 12 months. And you can see the different study arms, with these colored ones being the different interventions and the blue ones being the control. So you can see that at 6 weeks, nobody had systemic inflammation. I think this one up here was a clear outlier. We've removed it. Nobody had systemic inflammation at 6 weeks. But from 3 months to 6 months and then persisting at 12 months, the controls clearly developed systemic inflammation. In very marked contrast to that, the average levels of AGP really didn't change in the intervention arms. So we pretty much prevented inflammation almost completely. And it didn't matter which study arm you were in, systemic inflammation was avoided. And that seemed to occur even though we stopped the intervention at 6 months. So they were here at 6 months. We didn't see them until 12 months. And yet at 12 months, they still had low systemic inflammation despite no intervention. Of course, some of these children leaked into the inflammation arm, but really very few. This is obviously just a laboratory outcome, but the difference between the controls and the intervention is actually greater than the difference that Prendergast reported between stunted and non-stunted children in Zimbabwe. So we think this is already an important difference in inflammation. So this, we feel, is a good early start for these babies, and I'll come back to that later. If we just look at the proportion of children, babies in the first year who had inflammation, around 40% at 6 months and 12 months in the controls, but very few at 6 months and certainly less than 10% at 12 months. And of course, highly statistically significant differences. If we look at the time course of systemic inflammation in the controls, this is quite interesting. These are the different time points: 6 weeks, 3 months, 6 months, 12 months. The orange is the ones with raised inflammation, and the gray are the normals. So two immediate observations. Obviously, there's a group of control children who didn't get any intervention who did not develop inflammation, and they remain normal up till 12 months. But also about a third or so developed inflammation by 3 months, and they tended to persist. Once they got inflammation at 3 months, they tended to also have it at 6 months and again at 12 months. And then there are some in the middle who gained and lost inflammation. So, you know, this is the 40% by 6 months, and the more or less 40% by 12 months. So this relates to the sort of typology of inflammation, and there might be subsets of children, some resistant to systemic inflammation, others susceptible, and this starts in fairly early life. So that's something we're thinking about. This is the same plots, but with the intervention arms. Clearly, much less intervention, much less inflammation in the intervention arms, as already said. But also, what we found was, amongst the children at 3 months in the intervention arms that did have inflammation, they were more likely to resolve by 6 months than the controls. And that was statistically significant. So that suggests that the interventions not only prevent inflammation, but if it's present, they might be good at resolving it. Having said that, if we then look at 6 months to 12 months, that was also more or less true, although borderline statistical significance, that the intervention arms with inflammation at 6 months tended to resolve more than the control arms, even though we weren't giving the intervention. So that suggests that if you get healthy gut and obviously a healthy immune system in the first 6 months, that has a carryover effect that can help to resolve inflammation even when it occurs. So two possible different outcomes from these sort of Sankey plots. I've just run through a few of these similar charts. So we've talked about chronic systemic inflammation. This is acute inflammation, plasma C-reactive protein. You can see immediately that probably about 50% or more of all children at all time points had a raised CRP. And this is probably related to very frequent infections, possibly mainly viral infections in this population. But nevertheless, the intervention arms were still better off, certainly at 3 months and 6 and 12 months, than the controls. So that's acute inflammation in the blood. So we feel we've established kind of proof of concept against our original hypothesis that prosymbiotic administration in this population reduces systemic inflammation. Then let's look at the gut, the biomarkers in the gut. This is the fecal myeloperoxidase. This is the gut inflammation. And again, here, we can see that there was evidence of inflammation even at 6 weeks in all the study arms. And most children had some gut inflammation at 3 and 6 months. And it was certainly a lot higher at 12 months. So our intervention has definitely not completely prevented gut inflammation. As I stressed at the beginning, we only gave it once a week after the first 10 days. Possibly, if we'd given it more frequently, we would have reduced gut inflammation in the intervention arms. We certainly did see it did reduce gut inflammation because we have some significant differences between the intervention arms and the controls. So maybe increasing the frequency would have given us a better outcome for intestinal inflammation. This is the gut leakiness, the alpha-1 antitrypsin leaking out into the stools. Probably less abnormal results until you get to about 12 months when 50% or so of children have got abnormal leakiness of the gut. Pretty similar in the study arms, but definitely better in the intervention arms than the control arm at 6 months. So certainly at 6 months, possibly a little bit at 3 months, the interventions seem to have improved gut leakiness, reduced it. And then intestinal fatty acid binding protein, this is the enzyme protein that leaks out of mucosal cells when they're under stress. Again, mainly normal values, which is good. This was the only marker of an early difference at 6 weeks, better off in the lab 4B symbiotic arm than the controls at 6 weeks, but that's probably borderline significance. But interestingly, we saw probable differences at 6 months, although not significantly differently, but certainly different at 12 months. So less damage to the mucosa in the intervention arms, certainly at 12 months than in the controls. Interestingly, we also looked at stool pH. Having a lower pH is a good thing. We say between 4 and 5 would probably be thought of as being healthy, because a low pH probably deters pathogens, so is beneficial. And most people would say that a major benefit of probiotics is that they result in the production of short-chain fatty acids, particularly butyric acid. And this is very healthy for the gut, both in terms of gut development, immune development in the gut, and anti-pathogens. So, and of course, organic acids make the stool more acidic. And we really couldn't see any difference in pH, apart from a possible difference at 6 months here. We really couldn't see any difference. So our conclusion is, or just based on this stool pH, is that probably production of short-chain fatty acids is not the reason, was not a major mechanism whereby gut health was improved by the pro and symbiotics. If we look at how these different gut health biomarkers relate to systemic inflammation, in univariate analysis, no relationships at 6 weeks, but at 3 months, we can see that worse mucosal integrity was strongly associated with systemic inflammation. I think 0.3, 0.24 are quite high correlations and certainly statistically significant. At 6 months, it was gut leakiness that seemed to be related to systemic inflammation, and at 12 months, it was all three. Clearly, these biomarkers are interrelated. So if you have gut inflammation, you will also have some gut leakiness. So when we did a multi-regression analysis and put all the three biomarkers together, nevertheless, we still saw the same sort of pattern. Although the associations are weaker, but still highly statistically significant. So we think we can expand the proof of concept to say that prosymbiotic administration improve gut health, whatever that means, that's structure, function, immunology, inflammation, and thereby reduce systemic inflammation. Of course, prosymbiotics are given orally, so any effects in the blood, I guess, have to be mediated through the gut and therefore could be captured by improved gut health. Okay, so we also looked at growth factors, insulin-like growth factor 1 and its binding protein 3. And as opposed to our other biomarkers, the higher the value of these growth factors, the better. And we can see here that IGF-1 was significantly better in at least 2 of the intervention arms than controls at 6 months, and IGF-BP3 at 3 and 6 months. So through improved gut health and possibly reduced systemic inflammation, levels of growth factors were higher in our intervention arms, at least at those time points. This is just a very quick summary of all this biomarker data. So here's the 4 time points. Reduced systemic inflammation in all the intervention arms from 3 months onwards. Reduced acute inflammation, at least at 3 months and 12 months in all the intervention arms. Reduced intestinal inflammation in at least some of the intervention arms at 6 and 12 months. Reduced leakiness in all the intervention arms at 6 months. Better integrity in one arm at 6 weeks and in all arms at 12 months. And better growth factors at 3 and 6 months. So I think in all these analyses, there are, I think, around 30 statistically significant differences between the intervention arms and the controls. In every one of those 30 analyses, it's a beneficial effect in the intervention arms. None of the differences suggest an adverse effect on these laboratory biomarkers. If we look at mortality, it was fairly equal across the intervention arms, and certainly no higher in the intervention arms than it was in the controls. We had quite a number of serious adverse events, as perhaps is not surprising in this population, 102 over 2 years, mainly pneumonia, malaria, anemia, gastroenteritis. All these were looked at, none were attributable to the interventions, and they occurred in a similar way across the 4 intervention arms. So no evidence of reduced morbidity, but also no evidence of adverse effects. These are minor adverse events by the study arms, and again, they were all very similar. What you can see here in the overall, there was 8,675 adverse events recorded in these 600 babies over 2 years. So that's about 14, on average, 14 per baby. So this population had lots of acute infections and other minor adverse events. They'd have received a lot of support for these issues from the study team, including referral to clinic, medicines where these were required, but similar in the 4 study arms. I just want to now focus on growth. I can see time is getting on, so I'm getting to the end. So this is a well-known analysis of growth profiles in poorer countries compared to the WHO standard. So I'm sure everybody here is familiar with the WHO 2006 growth reference. This is this green curve here. So this is what WHO would say is optimal growth. This is what babies who are from poorer communities, but nevertheless are raised in optimal circumstances, exclusively breastfed, mum didn't smoke in pregnancy, they're not a twin, they're not low birth weight. This is what they should grow like. So around a 0 Z score, this is what generates the reference, and between -2 and +2 standard deviations, of course. And then populations such as the population of children in Kenya would be shifted to the left. The whole of the population shifted to the left, whether you're looking at length, weight, mid-upper arm circumference. So we expected, of course, our controls to look like the shifted to the left population, and we expected, with any luck, our intervention groups to be shifted to the right towards the green line. And we had a very surprising finding. So here are our anthropometric indices at 6 months. And you can see that essentially, whether it's length for age, head circumference, weight for age, arm circumference, they all look like the WHO reference. So they're all around 0 and pretty spread between -2 and +2 standard deviations. So, and that includes not only the intervention arms, but also the controls, the blue line. So on the one hand, we've achieved really excellent growth in these children in the PROSYNK study. And as far as I'm aware, this is the only intervention study that has normalized growth in children in a poor community. Obviously, lots of different intervention studies, lipid supplements, WASH, micronutrients, pregnancy interventions. I don't think any study has normalized growth. But of course, we cannot subscribe this to the interventions because the controls also grew well. And that's why I've been emphasizing the great support to the participants through the study. We presume this is an effect of being in the study and the support that they've received. But nevertheless, these children in this study have achieved very good growth. And I'll come back to that very shortly. These are the same sort of indices at 12 months. Again, all looking like very good WHO standard data. This is weight gain at the different time periods in the study arms. Optimal weight gain, 300 grams per week initially, tailing off a bit as you'd expect, but no difference between study arms. This is height gain, same picture, very good height gain over the first 12 months, no difference between the study arms. Very quickly, we used a cold chain to deliver our probiotics, symbiotics to the households and gave the doses under supervision just to make sure that the bacteria, the lactobacilli, bifidobacteria, were alive and kicking. And we did quantitative counts to confirm that when they were delivered. But in fact, when we didn't use a cold chain and we just kept the products on the shelf in the laboratory, we measured the temperature between 20 and 30 degrees centigrade in a sort of diurnal cycle throughout the year. In fact, the viability of the labinic dropped pretty quickly. Of course, we've got labinic formulated in a powder, not in the MCT oil, which is normally used, because we wanted it as a symbiotic. But the Lab 4B did very well. There was some drop off, but then they maintained viable counts for almost up to a year. So we know we can, if we think we need to, we can increase these counts by putting more bacteria in the capsules. That's fairly simple. So we think this intervention could be delivered quite happily without a cold chain. We did a couple of add-on projects. We did a qualitative study, and suffice to say that this study has very strong support from the mothers and the families and the communities that have been involved. They think their children have done really well. And of course, they're right, looking at the growth curves. They think their children have done really well, and they're very receptive to the intervention and supporting this study and future studies. We also looked at we did quite a complicated cost and implementation feasibility study, thanks to my colleagues in the Liverpool school. And to cut a long story short, without a cold chain and delivering the probiotic powders directly into the baby's mouth, The cost is, with delivery in the community, is probably about $9 or so per infant for a 6-month course. That's the 32 doses we gave over 6 months. So we think this is quite affordable. Let me, sorry, get rid of that one. I can. So we think that administration of a live multi-strain bifidobacterial lactobacilli for the first 10 days, weekly to 6 months, reduces systemic inflammation, improves gut health. The effects persisted. So we feel if you get off to a good start in the first 6 months, they persist. This effects persist. We didn't have any adverse outcomes, and we feel this is applicable to community level intervention as pragmatic and affordable. I just want to finish with a couple of slides saying how this work is going forward. We've been very fortunate in getting some funding through the Gates Foundation Collaborative Platform. So we're markedly increasing the range of inflammatory markers that we're looking at. In fact, 250, rather than AGP and CRP, we will have 250 inflammation biomarkers on all the samples at the different time points. We've also going to have metagenomic sequencing at a subsample of stools at 3 months to see the normal development of the gut microbiota and function, and also how it's affected with the intervention arms. And of course, we'll have support with fairly high-level statistical analysis because of these complex data sets. So that's working with colleagues in Stanford University. There's Iwaret and Alois from Kenya on a recent visit there. And we look forward to that information coming online. Clearly, we have the ongoing Stunting Hub project. Very interested to hear what the biomarkers of gut health and inflammation and nutrition will look like in this study across these 3 different countries. That should help us to refine the typology. The interdisciplinary analysis will help us to look at the pathways that affect gut health, including the microbiota, parasites, all the environmental variables and behavior such as feeding. And then, of course, we're testing one of the products used in Kenya in Senegal. So in West Africa rather than East Africa, and we'll be very interested to see if we get the same results in a different population. I just want to finish with how we're thinking of developing the gut power concept. So although this is based on a single study, which of course we need to be cautious, we feel that we have a good foundation that we should develop this concept towards using a probiotic or symbiotic as a public health intervention for infants in low resource settings to improve long-term health and development. First and foremost, we think our PROSYNK cohorts that have had a good early start, we should follow them up at 5 years to see what their longer term outcomes are, especially in terms of organ development and perhaps early biomarkers of non-communicable diseases. We've clearly got 2 groups. We've got a set of babies with an excellent early start, good growth, reasonably good gut health and no systemic inflammation. That's the supplemented group, all 3 put together. And then we've got the controls who, although they had inflammation and not so good gut health, they certainly had good early growth. So we would plan to recruit some non-PROSYNK controls at 5 years, and that should help us to untangle the effects of inflammation and growth and gut health on these longer term outcomes. And of course, if we're proposing to take this to the community level, longer term follow-up to make absolutely sure there's no adverse outcomes, because we have changed the biology of these children, is of course very important. And then very finally, we feel that, again, to support a program of community intervention, large scale, we should probably do a randomized blinded control trial using just one product against a control, and perhaps give it more frequently to try and improve gut inflammation and gut biomarkers, and possibly giving it for longer, to give it up to 1 year and look at the type of outcomes, and then also establish these as cohorts for long-term follow-up, pops into later childhood and early adult life for organ development and NCDs. Clearly, all this work in Kenya was done by a very large study team. Big thank you to them, and thank you very much, everyone, for listening. That's the end. Thank you.
Great. Thank you so much. That was really, really impressive. Some great insights there. Whilst you were speaking, we had a few questions, and I know we're pressed on time, so I'm just going to go ahead. Hopefully, we can get through all of them. So the first one we have is from Natalia, and she would like to know what are some common misconceptions about gut health and its role in preventing malnutrition and stunting that you may have encountered in your research?
Misconceptions. Yeah.
Yes.
Okay. Um, yes. So I think there's at least within the academic community, there's very wide acceptance that this poor gut health, characterized as environmental enteric dysfunction, clearly contributes to poor growth through malabsorption, through growth hormone deficiency, and longer-term poor outcomes. I think that is very well established. There's multiple studies that look at those relationships. What we haven't had yet is an intervention, and I think that's where our study comes in. In terms of misconceptions, probably that's known in the academic community, but we need to get that out into the NGOs and the policymakers. But I suppose a game changer for that is having an intervention that they can pick up on. In terms of obstacles, the mantra of exclusive breastfeeding is clearly a problem, a potential problem, because many people would say if these babies were only exclusively breastfed, all would be well. Breastfed to 6 months, they'd have very healthy guts. What we know from several studies, Zimbabwe, Bangladesh, our study, is that these babies get poor gut health even when they're exclusively breastfed. So, giving a supplement to babies who are otherwise exclusively breastfed, again, is a barrier that needs to be crossed. And, these babies need extra help. And even if that means giving them something by mouth when they're supposed to be exclusively breastfed. We give vitamin A, we give medicines. Perhaps we should also be giving a probiotic or a symbiotic. I don't know if that answers the question or some of it.
Yeah, no, I hope so. I think she'll probably put something in the chat, but yeah, hopefully it does. So the second question that we have is in terms of policy and practical applications, how can the findings from your research be integrated into national health programs to combat childhood malnutrition more effectively?
Yeah, so that's a very good question. At the moment, I think you'd have to say they can't. We've got one study essentially, and we've only got biomarker outcomes. We've only got good things in the laboratory so far. We haven't got actual health outcomes. As I say, this is probably, we think, because our controls did so well. And this is where the Seng study in Senegal could really kick in, because I think in the Seng study, there's been less intervention, less support. And therefore, if we see the same health benefits in the Labinic symbiotic group in Senegal, but the controls do less well, there we might see some health benefits. So I think to get this into policy, we need to demonstrate health benefits. Having said that, if I was a mother in Western Kenya and I was offered a probiotic for my baby, I would give the baby the probiotic based on the results from this study. But I think we need to, follow on research, and maybe this is where the Senegal study will kick in to show some definite health benefits.
Thank you very much. This question is actually I think you partially answered it, so I'll ask it anyway, just for the viewer. But they wanted to know, looking ahead, what would you say are the next steps or future directions for research in the area of gut health and its role in preventing malnutrition?
Yeah, very good question. I think the first thing is we've got to look broader than just malnutrition. Clearly, being underweight is a problem, so that's true. Being a centimetre or two shorter, is that really a problem? So I think we need to obviously keep stunting as a marker of disadvantage. That's very helpful, because we know if we go into a population that's got a lot of stunting, we know they're disadvantaged. We know they'll have poor gut health. So that's very useful. But in terms of outcomes, we need to go beyond just linear growth. We need to be thinking about brain development, lung development, kidney development. That's why, and so possibly the biomarker net needs to be much wider than just the gut. So as I say, we may be able to do a lot in ameliorating the adverse associations of stunting without actually addressing stunting itself. So having said that, we also need to learn more about the gut. You know, the immune pathways, the inflammatory pathways, how this links with the brain, how this links with the lungs and other organs, and therefore a broader range of biomarkers looking at the gut would also be helpful, so we can make sure our interventions target the pathways that is linking the gut and the brain, or the gut and respiratory function, for example.
Great, thank you very much. So we have 5 minutes, but I'll keep on going. So the next
I guess as part of that answer is, as always, you know, we need a lot more research, which means it's good for us. We can stay in jobs, a lot more work to do, a lot more funding to win. So, and you know, a big topic area needs a lot of different disciplines to come in and look at this. Yeah.
It's so as for the next question, firstly, they wanted to thank you for your presentation. And then they would then like to know, while discussing the gut microbiome, did you come across any research on curcuma? I'm not familiar with the term, curcuma zedoaria, for promoting gut health in children. They noted that it's a common ingredient in Indonesian jamu, which is a traditional drink used for digestion.
Yeah. The answer to that is no. I don't know of any studies looking at gut health malnutrition in that context. I think in the UK it's coumarin. I know that's been looked at for esophageal health in certainly, I know, you know, I was previously in Swansea University. I know they were doing research on coumarin, I think, and reflux esophagitis, and you know, degenerative problems of the esophagus. So I think there's quite a bit of work in that area. And again, that's an interesting, you know, that's another, from my point of view, a very interesting potential intervention. It's, you know, it's probably safe, it's probably cheap, and it probably won't cause antimicrobial resistance. So, you know, like an antibiotic would or something. So again, that would be an interesting area to look at. Yeah, but I'm not aware that anybody's done anything on that in this particular malnutrition gut health area.
Thank you very much. So the next question we have comes from Rachel, and she would like to ask your thoughts on whether you think Western marketing is a move to sell us probiotics, and whether that kind of helps or hinders this sort of research that you're currently doing.
Yeah. Okay. My own biased view about, I've done quite a bit of research on probiotics in different areas. My own view, and I think this is where we've scored with the Kenya study, is that there's a window of opportunity in early life when the infant's gut microbiota is not well developed. So I think that is an opportunity to put in, to modulate the gut microbiota, to put in lactobacilli bifidobacteria. But that's probably an early window of maybe the first 6 months, as we've done, maybe the first year. After that, once the baby has developed a fairly extensive gut microbiota, that then becomes very stable and hard to modulate. So I think just as we've put bifidobacterial lactobacilli in to try and give colonization resistance against pathogens, if you wait too long, the gut microbiota will give you colonization resistance against probiotics. So you take the probiotic, but the normal gut microorganisms just fight them off and they don't do anything. So my own view is that there's I think of a lot of the research that's been done for probiotics in older people, is very underwhelming. They tend not to work. My own view is that this window of opportunity in early life when they may really have an effect. In terms of, is the commercialization, because it's a huge industry, is that beneficial? In a sense, yes, because the more money the probiotic companies have got, the more likely they are to support research. And so it's, you know, for probiotics to have a good profile about being healthy, well-being, and for there to be plenty of money. And, you know, if they're produced at scale for rich countries, that presumably means that they're cheaper to deliver in poor countries because they're already very widely available at scale. So one of the suppliers, you know, Coltec in South Wales, who provided us with the Lab 4B that we've used in our studies, and used in other studies, they do a lot of research around their probiotic consortia. And that's really helpful. It builds the research around specific products. And so the more probiotics they sell in Boots, because they're for selling Boots, their probiotics in the UK, the better, because they will then do more research. There we go.
We do have a few more questions. Are you okay to stay on for an extra 5 minutes?
Yeah, sure. No problem.
Thank you very much. So I'll go to Kevin. Rachel had a few more questions, but I'll go to Kevin just for sake of fairness. So he wanted to know, is there a link between gut health and behavioral issues or mood in children? And secondly, how does gut health relate to conditions like allergies, asthma, or eczema?
Okay, I'll answer the second question first. For the behavior, I wonder if Iwaret would comment, because this is definitely her area. And but in terms of allergy, yes, there's a big field of research about probiotics to prevent allergy in babies and atopic diseases. And you know, this is either giving them during pregnancy to the mum, or just giving them to the baby, or both. And in fact, in Swansea, we did a study called Probat where we actually did that. We used the Lab 4B probiotics that we used in Kenya, gave them to newborns in Swansea, South Wales, and indeed we had some effect on preventing atopic sensitization. So I think the data in meta-analysis is still a bit here and there, but nevertheless, that is a big area of research. Iwaret, are you able to come on and answer the question about brain development and behaviors?
Yeah, I'll just give her the access. Should be able to talk. Oh, I can't hear her.
Can you hear me?
Oh, yes. Yeah, we can.
Okay, great. So I'll just try to answer that question about gut health and mood and behavior. There's currently a lot of studies on neurobiotics, on the interaction between the microbe and on mood and behavior. It's being done a lot in adult studies to try and see whether it's helps prevent neurodegenerative disease, a few studies on autism, but it's not really a clear pathway, although we know there's a direct link between the gut and the brain, which is the gut-brain axis, a lot of signaling happening through the sort of endocrine system and the enteric nervous system. So there's a lot of work that is going into the gut-brain axis kind of studies affecting mood and behavior, but I can't say conclusively that there's a direct answer that it affects mood and behavior, but a lot of work is actually going into that.
Great. Thank you very much for taking time to answer that. So just final questions. So Rachel also wanted to know, could you tell us a little bit more about the process of data collection and how the participants felt about participating in the actual research and whether there were any challenges in doing so?
Yeah. Yes. Okay. So again, Maritu is on the line. Who's done the Senegal study. I don't know if Maritu, you're following and are able to answer about what the mums think of and the families think about these capsules in Senegal. Are you able to tell us, Maritu?
Yeah, I've given you permission to talk if you'd like to answer that. Is
failing that, Iwaret could certainly talk from the Kenya perspective. So I don't know if you want to come in first, Iwaret.
Sure. Can you still hear me now?
Yep.
Yeah.
Okay, great. So I think when we were doing our dissemination, and Prof. Steve, you were there was actually sort of like positive response from the participants, from the mothers of the participants who were in the study. The major challenges we got early on was because of the myths that came about with how we were conducting the study. The frequent visits at home, the frequent contact, it was not so common. So there were a lot of myths
Great, thank you very much. I'm not sure if Maria too wants to come back. So just we have one final question. If you've got to be someone, that's totally fine. So
No, that's okay. Last question. Go for
okay,
it.
great. So the last question is, could you elaborate on how this research is being carried out in the Action Against Stunting Hub, and is it different, and if so, how?
Yeah. Again, Ben is online, and if Marito and contact Ben's online, do you want to say some of the differences that in the Senegal study, Ben?
Ben, I've just given you access to talk if you'd like to.
Thanks, Steve. Just to mention in terms of the behavior of the mothers in Senegal. So one good thing is that they perceive malnutrition in general as a problem, and so they really welcomed they were really keen to participate in the study. They joined massively, and we had situations in which even those in the control arm were disappointed because they didn't receive the symbiotic, noting that it's an open study. In terms of differences with what we are observing with the study in Kenya, not really much, just mainly in terms of practicalities of practical considerations in terms of implementing this kind of study, notably receiving some regulatory approvals, administrative authorizations. The research landscapes are not really the same. And so you can imagine that in terms of implementing this kind of study in the setting of West Africa wouldn't be the same as in East Africa. So those are the main challenges. Also, I would just like to mention there are important geographical issues, notably the climate, etc., that do make the implementation of this kind of studies difficult. Extremes of temperature, huge rainfalls, floods, etc., that make field workers have difficulties implementing this kind of study. Thank you.
Thank you very much.
Just to add to that, as I've said, I'm hoping that the Senegal study might show a bigger effect of the intervention, not because it'll be better in the intervention arm, but because the controls will have done worse than the controls in Kenya. Because I think it's in the Senegal study, it's in the health centers are quite disparate. I suspect the study team for the Stunting Hub has not been able to support the babies as well as was done in Kenya. Therefore, their growth, their morbidity is probably more like normal than our controls in Kenya. So we may see a bigger impact to the symbiotic because the controls didn't do as well. But let's see.
Great. So that actually concludes all the questions. Thank you very much for staying on a bit later to answer them, Professor Allen. Much appreciated. With that also concludes the session. If anyone wants to watch a recording of this, that will be made available later this week on our YouTube channel, and we will send an email to the accounts that you used to register with the seminar for a link to that. So yeah, that's it, guys. Thank you very much, everyone, for attending.
Fantastic. Thank you, Dylan and Chelsea, for setting this up. Much appreciated.
Thank you very much.
All right. Bye-bye now. Thank you.